Hello, in this video we're going to be talking about the electron transport chain and chemiosmosis. Let's not waste any of your time. Let's get right to it. Now when we're thinking electron transport chain and chemiosmosis, the thing we want to think about is one, the location. It's on that membrane as we looked at in the earlier video, on the far side. Glycolysis started in the cytoplasm at the top or above the mitochondria. If you're looking down upon it like at a map, it'd be on the north end of the picture. And now we've moved all the way through the middle, and now we're on the far side, more the southern end, if you're looking at it like a map, on that membrane. But you can see the components. We're talking proteins. Okay, these are going to be pumps like we've talked about in the plasma membrane during active and passive transport, and these are going to be active transport. And because if we think in active transport, we want to remind ourselves that means that one, ATP is involved, okay, but also two is that we're moving elements, okay, hydrogen in this case, against the concentration gradient. We're going to be moving it to an area where we have a whole bunch. We're going to be moving some into an area where they have a whole bunch. So that's the idea of active transport. So keep that in mind. But it's the idea of all of these H's that have been dropped off, okay, by the electron carriers are now going to be moving, processing through, with the idea of generating ATP molecules. And when we get to chemiosmosis, as we talk about, that has a special protein pump. ATP synthase is what it's called, which means making ATP. But we're going to see where the NADHs drop off their H's, where the FADHs drop off their H's, and the role that water plays, and then of course our pump at the end. Okay, in this diagram, we're just going to remind ourselves where we are and where we've been. We started with glycolysis outside in the cytoplasm. We moved inside that membrane to do pyruvate oxidation. We moved into the middle of the large area for the Krebs cycle, and now we've moved across, and we're on that membrane in the bottom of the diagram. Okay, so electron transport chain and chemiosmosis. Again, it's in the membrane, and it's a series of proteins that are going to process these H's with the idea of making ATP. Okay, and each pump that we're going to be looking at, has that protein gradient. In other words, the H's, once they remove their E's. Hydrogen only has it's got a ATM mass of 1, so it's only got that protein, got that electron, and so all it's going to be that protein. But each protein is going to transfer electrons to the next carrier. In other words, moving it down the assembly line. But we'll look at that. Electrons end up on oxygen. Okay, oxygen is reduced to form water. But we're going to showcase all of that stuff in the next slide. Alright, so here's where we want to kind of begin to focus on what's happening in individual places. So we're going to look at this diagram. Obviously you can see 4 blue objects. Those are each protein pumps, And so we're going to start here on the far left with the first one, okay, and we'll just name it the protein 1. You can see the NAD above it there. That's the protein that NADHs drop off their H's. Okay, it's a specialized pump, as we've seen through all of this talk about proteins in the membrane, specialized, and NADs, H's have their own particular spot, and they drop theirs off, nobody else does. Okay, the FADHs, okay, their H's run through the second protein. So we've got the NAD's got their own, FAD has its own, and then that third one, okay, is where oxygen comes into play, which makes this an aerobic respiration process, because some of the H's are used to form water. Not all of them, as we're going to see. A bunch of them are used to make the ATP molecules, but there are some that are used to make water. So you've got the oxygen shows up, and then it grabs 2 H's, and a water molecule is formed. That happens at the third protein. Okay, the fourth protein, the one on the far side, and our next slide we're going to look at a bigger version of that, bigger picture of that, so we can really break it down on what's going on, because it's a pretty special protein pump. But essentially what happens is that the H's from the NADs, the H's from the FADs, and then the H's that aren't used to make water, they all move through their pumps, and then they parallel down to, process down to the ATP synthase. So they're essentially, they're inside that inner membrane, they move across the membrane, now they're in that outer section, basically right up against the plasma membrane edge of the mitochondria, and they'll move down, and then they'll re-enter the inner membrane area, going back through the specialized pump called ATP synthase. So here's the ATP synthase, a bigger picture so you can see, and what you see is a pretty spectacular protein that has a lot of what would seem to be simple machine kind of parts. The other thing that this brings to mind, that when we looked at these complex, even though they're super small, I mean, this is one part of the membrane of the mitochondria, which the mitochondria is microscopic, and so one little bit part of that would be super small. But anyway, when we looked at flagella, we also looked at an object that seemed to have several simple machine parts in order to make it work. But you can see here in this ATP synthase, the rotor at the bottom. Again, you can kind of see a wheel and axle thing going on, and what looks to be the microphone coming out the side, but that's not what that is. But anyway, you can see the working part, the rotation at the bottom, the rotation of the stem or the stalk, and all this stuff happening. But essentially what happens is that the ATP molecule, the H's move through, okay, and when that does, it causes the machine to crank, and the energy is generated to be able to stick that third phosphate onto the ADP to make ATP. Well, that is our video. When we come back next time, we're going to again be looking at the electron transport chain and chemiosmosis in video number 2. We'll see you then.